Hello and welcome both to those who are here and those who are online. My name is Alan Love. I'm director of the Minnesota Center for Philosophy of Science, and it's my great pleasure today to introduce Pamela Reinagel, who is coming to us from University of California, San Diego, and the Department of Biological Sciences in the section on neurobiology. Before I say a little bit more about Pamela, I want to highlight, so for those who are not aware, that this talk fits within a broader initiative that a number of us have been working on for the last several years called the Many Faces of Reproducibility. You can find out more about those events that we've had in the past, some recordings of those things online, as well as future things coming up by going to reproducibility.umn.edu. I just wanted people to sort of be aware this is something that we're broadly interested in here at the University of Minnesota, and we're delighted that Pamela can come talk to us about these sorts of things. So I met Pamela last summer online when we were at a virtual conference, and got to talking and realized there were these mutual interests. And so that began a conversation that has led to this opportunity today. Pamela is currently on sabbatical and thinking through these issues as well as others during that time. And so today, what she's going to share with us is a practicing biologist perspective on reproducibility in science reform. Before I let her do that, though, let me highlight to those who are here and those who are online that there will also be an event tomorrow morning where we will read a paper with Pamela together in the biology interest group. And then there will be a lunch at the campus club talking about a paper of Pamela's on the topic of NHAC. If any of those things are of interest to you and you don't have information about how to get there or the Zoom links and the like, you can either email me, aclove@umn.edu, or mcps@umn.edu for those details. That's enough from me. Now, Pamela, please.
Thanks, Alan. I'm so glad I was able to make it here. It was starting to look like COVID might prevent this from happening, but it's wonderful to be here in the format that we can be. To just introduce myself a little bit and why I got interested in this topic, I am an experimental biologist. I have been for essentially my whole life. I've been working at a bench in a lab for probably just about 40 years now. And I got into science, I got into biology specifically because I just thought the way of knowing of experimental biologists was so beautiful and inspiring. Because I actually considered going into the history of philosophy of science early in my career, I decided that I would learn more about how biologists do this thing by actually doing it. And so I've been busy doing science for all this time, and now I'm sort of circling back around to revisit the question of what is it that people are doing in the lab to discover, what is the kind of knowledge that we're generating, what is the process by which we generate it and validate it, and these interests got rekindled by the emerging reproducibility crisis and this big conversation that's happening throughout the world about whether or not science is reliable, how can people know if science is reliable.
Pamela, can I have you stop for just a second? Turn off the mic and try and just talk forcefully.
Okay, is this working for people as far as sound?
Can I get feedback from people online if they can hear Pamela better? Go ahead.
Is this, if I talk like this, is this going to work for people? Yes?
Okay, I think that's better. Yeah, okay, better on the audio quality.
Too much mic, great.
Okay, yep, good.
Excellent, thank you for catching that. So, so as these papers started coming out, like these ones that I'm sure you're all familiar with, why most published research findings are false, or undisclosed flexibility allows presenting anything as significant. Of course, I was very concerned, and I started reading them, and I will say, I think that these papers started a very valuable discussion, and I agree fundamentally, sort of with all the mathematical claims that are in these papers. I think it's very true that the issues that they raise carry the challenges that they carry. But when I started trying to translate that to my research and my mentorship and my teaching, we kind of struggled to understand how do these arguments apply to research in biology. And it seemed there were many cases in the kind of biology, at least that I have seen in my career, that just don't fit the picture of how science is done. And so I've been taking this sabbatical year to talk to philosophers of science, to people in education, and to people in statistics, to sort of try and frame a more biology-centric agenda for science, reproducibility, and reform. And I should say right now, biology is a huge, huge discipline. There are many branches of biology, and I can't speak for all of them. Very notably, the translational and medical fields like clinical trials or epidemiology, very different from what I'm going to be talking about. I'm only going to be talking about sort of experimental model system laboratory biology. And so I will say biology loosely, but mostly as a shorthand for particular style of biology. And I'm going to give you some concrete examples so you know which ones I'm talking about. Okay, so we start with this idea of the scientific method, as if there were a scientific method that we all use. And most of the presentations, actually, is there a laser pointer? I'll just keep going, but if you have one. So most of the presentations that you'll see, at least at the elementary level, will explain that the scientific method works this way. You come up with a hypothesis. Apparently that can just come out of the sky, from nowhere in particular, or it doesn't matter where it comes from.
The very top one should be the laser.
Very, very faint. Okay. I think that may not help enough. I'll just keep going. Right, so you come up with a hypothesis, it doesn't matter where from, and then you rephrase that as a null hypothesis. So, maybe I think that, oh gosh, I have to make up fake hypotheses now. Maybe I think that it always snows on Thursdays in Minneapolis, and then I turn that into a null hypothesis that, it is not more likely to snow. You know what this story is. And then we design some experiment that could falsify that null hypothesis. We make predictions from that hypothesis that we can then test. And then we collect data. And the main feature about data that's usually talked about is that you have to sample some populations. There's some large population you want to generalize about, but you can't measure all of the members. So you measure some and you generalize. That's the main feature that gets emphasized in the treatments I've seen. And when you're done collecting all this data, you perform a statistical test. And then you draw your final conclusion. So, for example, if your p-value of your statistical test is less than some threshold, like 0.05, you say you've proven you have an effect. And if you have a p-value that's larger than that threshold, most of us have now realized that you're not supposed to say you have no effect, but you can say you have no evidence for an effect if you don't cross that threshold. So this is the story. And a lot of the discussion about the reproducibility crisis and investigations in metascience, trying to characterize like whether people are doing this right, sort of predicated on this idea that there's this ballistic event in which a hypothesis gets framed and then a bunch of things unfold sort of inexorably until you get to the end and then you have a conclusion. And then we talk about like whether this conclusion should be considered reproducible or reliable as a measure of whether this enterprise was a success. And as I talk to people about these things, I just got feeling baffled by the fact that I've never seen anything like this in any lab in my life. So I found that by talking to people in other disciplines, often were a little surprised and were interested to hear my just introspective generalization. If I tried to take a big step back from 40 years in labs of all different flavors, what I would kind of characterize as sort of the common thread of how we do stuff. And so that's what I was here to talk about, and then bring it back around to what implications that might have for how we should approach these science reform issues in light of that. Okay, so this is a tour of my personal experience of how experimental basic research works. And I'm going to start with my sort of cartoon characterization that's very abstract. Then I'm going to give a couple of really specific examples. I'm a little worried that it might be too much content to really try and follow those examples, but at least it'll give you some idea what kinds of things I'm talking about, and then come back out to the big picture again. All right, so the way I would describe the process that I know is that an individual researcher is living in a very tightly interpolated loop, which you can enter at any point. Maybe you make some observation that you didn't expect, and this causes you to change your view of how the universe works. And so you will create or update some kind of a model, a mental picture of how things work that could be consistent with what you've observed. And that'll generate an idea for an experiment, which is either to fill in some hole. You try to draw that model and you realize there's something you don't know what to put in that box. And so you need another piece of data. Or you think you filled in all the boxes and now you want to know if you got it right. So you do an experiment to see if the predictions of that model hold up. But the difference between this and that kind of ballistic model that I was talking about is that this loop is not happening over the arc of a project. So when you read a paper and they say, we showed X, P less than so-and-so, it's not one pass through this, it's like 1,000 passes through this. And one of the things I've been doing on my sabbatical in a very informal way, I think it would be really cool to do it formally, but I just don't have those resources, is interviewing scientists in different branches of biology and just asking them questions. What do you do? What counts as a result in your field? How do you know if it's true? And so forth. And one of the people I interviewed who is in developmental genetics, I asked them what they thought about peeking at the data. While you're collecting the data, do people peek? And she said, peek? They pore over it. That's what I try to teach them to do. They're supposed to be data whisperers. Every data point while it's being collected, you stare at it, like a parent stares at a newborn baby, and you just try and understand it. And that's what causes the next experiment to be a useful one. Right, so, and then these individuals are embedded in some community, maybe a laboratory where there's many people working on related projects, and through their one-on-one interactions and community interactions, like lab meetings or seminars, they pull together, the observations they've made, the experiments that they've thought of, the models that they're working with, and they try to hash them out in this space, common workspace, to try and come up with a worldview that sort of holds together for everybody. And at that point, you're usually dealing with, lots of different observations, lots of different working models. And so it's this sort of fluid, pluralistic exploration. And every once in a while, I've tried to indicate by some shaded out boxes, one of those observations, the group will decide didn't turn out to be reliable. It was anomalous, and we don't need to worry about that anymore, or just didn't turn out to be relevant. Or one of the kind of experimental designs we realize has a flaw. We're not going to pay attention to that kind of experiment anymore. Or one of the models is just no longer tenable, so we're not going to keep that one on the table. So things drop out, but that's not because they were considered to be flawed or bad contributions. They were part of the essential process of sort of building and winnowing a consistent worldview that's sort of ever more rich. And so there's no shame in being the person whose model got thrown out. This is the creative process that I see, in individual research and in labs. And then these labs are having that same kind of interaction with all the other labs in their field, where the yellow circle is now supposed to represent an entire research field. Certain labs have close connections, they might be next door, or collaborations. Others only know each other because they contribute something to this common space, which would be like conferences or journal papers. And again, in that literature, if it's a healthy field, there are lots of kinds of experiments, lots of kinds of observations, many active models that are being juggled and considered. And so these conferences and journal papers' main function is sort of an efficient way for this community to share and pool what they've got so far and what they're working on. And that's going to be important later, because I think the literature in the fields I've worked in is not intended to be a message to the outside world, here's what we know now. It's intended to help that lab you don't know in some other country who where they're working on a model you didn't know, your observation is crucial. So it's a communication within a community. So if you go back to this sort of ballistic one pass idea of testing a hypothesis, it's completely, it happens rarely, occasionally. It happens all the time in some fields. If you need to approve a drug or not, you better do an experiment like this. But when you're trying to figure out how the world works, this is rarely useful. So instead of framing hypotheses, I'm going to talk in terms of putting together a working model, which no one thinks is true, but everyone thinks is capturing some essential explanatory power. And I'm very aware there are philosophers in the room to who many of the words I'm using have very technical meanings, which I'll just say right now, I don't know those technical meanings. So if you think I mean something extremely specific by explain, for example, I probably don't. So that's just a disclaimer. I'm only just learning the vocabulary in your field. So when we design experiments, sometimes we're trying to test the prediction of a hypothesis, but I would say more generally, we're just trying to clarify what's going on. There's something that doesn't make sense. There's something murky, and we try and get more information about it. Sometimes that involves just getting a few more samples. I saw this weird thing in 3 mice. I wonder if I'll see it in more mice. But more often, you're not doing the same experiment again. You're thinking, hey, if some mice can do this, I wonder if mice can do that. And you do a new, different thing, not just accruing samples of some population. So you're doing either manipulations of your experiment or very targeted observations to get missing information. You do this tightly. And then you feel like you need to communicate this to the world when you feel like you've made some progress. So you made some observation nobody knew before, or you came up with a new twist on the model that hasn't been considered. And in my experience, nobody then sits down and does a statistical test. They sit down and they do a bunch of heuristic qualitative reasoning, bringing in all the background knowledge that they have to bring to bear on it. They try and think through whether this model makes sense and holds together. And that's the main thing that they communicate in their paper, in this discussion section, or as they explain their results in the results section, is their sort of heuristic or qualitative arguments from these observations to what it implies for a model. There is no final conclusion. It's never final. It's always a work in progress. And then you write your paper with your updated model or your new observation, and then some reviewer says, hey, is it statistically significant? So people go back to the bench and they say, okay, cram all those data, the pile we decided was useful, cram those data through some piece of software and spit out a p-value to satisfy some curmudgeon. And that's where the p-values come from, in my experience. And so right now, mea culpa. There's been an accusation of people in the social sciences and in biology doing so-called cargo cult statistics, going through the superficial motions without understanding anything about what it means. And I would say that most biologists are running around with coconuts on their ears. There's lots of superficial imitation of the appearance of statistics. And that is one of the culpabilities that I really think we need to do something about. Okay, so just to sum up my sort of brief summary of what we do, I think that the process, as I see it, is anything but linear. It's more some kind of a fractal dynamical system. In this kind of exploration, null hypothesis significance tests just don't apply. They're just not usable. But unlike the narrative that I see coming out of some of the social sciences, I don't walk away thinking, therefore, it's all bunk, it's junk science, nothing good is getting done. That just can't be true. I mean, the track record of biology in the last century is just stunning, the advances we've made in knowledge and understanding that have borne out in practical applications. I think there's obviously progress being made, but whatever is causing the success, it's something else. And so this is another one of my big themes, is I think that as a field, first of all, we need to realize that the null hypothesis storyline isn't honest and stop telling it. And we need to have much more insight, I think, into what our real inferential methods are. Because if it's all very implicit and uncodified, it's difficult to transmit that to the next generation of researchers. It's difficult to assess the quality of other people's work, and it's really difficult for the rest of the world to understand what they're supposed to do with the work that we're doing. So, okay, so that's my abstract thing. And then I thought at this point I should pause and give some concrete examples, because really when I say experiment, I could mean just about anything, right? So, and here's where I'm daunted, because to tell any story adequately would take more than the time we have. So before I put a slide up with an actual experiment, I'm just going to give this disclaimer. Those of you who know these examples are going to notice that I'm skipping tons of juicy details, super superficial description. Those of you who don't know these examples, if there are any, I will probably give way too much information that you couldn't possibly absorb. Right, so that's the balance I have to deal with. In either case, though, it doesn't matter, because all I want you to do really is take away, oh, when she says experiment, she means a thing like that. And I think I need to give two examples from two really different fields, so that you'll be able to pick out what's common more than what's particular. And so I want to go to the two fields in which I spent most of my life, the first being molecular biology, and specifically gene regulation, and the second example being visual neuroscience. And so since those are as different as they can be, and in my mind, exemplify very similar processes, I'm hoping we can pull away some generalizations. Okay, so here comes a whirlwind tour of some beautiful science that I won't be able to give justice to. So, so this is the bacteriophage Lambda, no relation to any other Lambda virus. Turns out Greek letters are used to name viruses. Anyway, bacteriophage Lambda is a virus that infects bacteria, and it can infect them in two different ways. The first way is called lysis, which is it injects this genetic material into that bacterium, which forces that bacterium to make 100 copies of itself until the bacterium finally bursts because it's so full of baby viruses and it dies. And they go off and infect their next victim. That's lysis. But the other possibility is called lysogeny, which is that the genetic information gets into that bacterial cell and decides to hide out in the genome harmlessly for any number of generations until the right moment comes in which it wakes up and lyses the cell. So it's a dormant virus. And so this was known already before even it was known that DNA was the genetic material. And it posed this great mystery, which is we've already formed this model, this worldview that says genes determine phenotypes. And we have these two bacteria that contain exactly the same viral genes, and yet two completely different phenotypes unfold. So how can that be? How can it be that the viral DNA or the viral genome in that cell, which is completely capable of lysing the cell, we know that because it's going to do it later, nevertheless doesn't do it. And at the time people took this on, it was not the case that people thought this virus was intrinsically important. Nobody thought it was clinically important. Nobody thought it was useful for engineering. None of that. So finding out the details of how this worked was not because we needed to understand Lambda. It was because people recognized it as a case of a general phenomenon that would be important to know. It's the same problem as how can it be that you have the exact same genome in your brain cells as your liver cells, and yet one's a brain cell and one's a liver cell, same problem. And they thought it'd be a heck of a lot easier to figure it out in a simple case like this. And the premise was that some general principles would emerge, and those are the goal of the research. And I think that's really important, again, when it comes to talking about whether science is delivering on what it's trying to produce, if you understand that what it's trying to produce often is not so much facts as concepts or principles. And of course, those are being built on facts, and those facts need to be valid, but it's a slightly different end game. Okay, so I'm going to jump into this story in the middle to just give an example of one experiment. So this would be like one paper from one person in one lab, one of those inner loops. So we're going to start at the point where enough research has already been done that they knew that there was a part of the genome of this virus. There were some genes over here on the right that are required for lysing the cell. There are some genes, there's a gene over here on the left that makes a protein called repressor. And whatever repressor is, it stops the lysis from happening. And then there's some bits of DNA in the middle called the operator, all shown in pink. And they don't make any protein at all, but they are necessary for this whole thing to work, for repressor to stop lysis or for lysis to come alive later. And so there was genetics and biochemistry and other things behind that. And so the researchers formed this model that maybe the repressor is a protein that actually sticks to the DNA. Sticks to the DNA and does something to it. I don't know, on lines that they didn't know, but does something that determines what happens with the genes. And so to test that, they set up an experiment. They purified some of this repressor protein biochemically. They purified another enzyme that can break DNA. And they had a method for putting a radioactive tag on DNA. And so they mix these all into a test tube together. And the idea is they tagged one end of this DNA with a radioactive tag. They put a little bit of enzyme in the tube so that every individual piece of DNA gets broken only once on average, but in all different places. And then they reason if this protein is sticking to this DNA, then it won't be able to be broken there, it'll be protected. And we'll know that because all the pieces that get broken at this location will generate a really short radioactive DNA piece. And the ones that get broken down here will generate a really long piece of DNA, radioactive piece of DNA. And we just won't find any pieces that are that length. And that's how we'll know, how we'll test our hypothesis. And so this is the experiment, the main figure from the paper from Johnson, Meyer, and Cashley, 79, in which they did the experiment. And in there's 8 lanes there. This thing's called a gel. It's like a vertical slab of jello. It's got little rectangular wells at the top, and you just take the liquid from these test tubes and put it in the wells, put an electrical current down it, so that the small pieces of DNA, the electrical current is moving the DNA down. Small pieces go really fast, so they get to the bottom quickly, and the big pieces are stuck up at the top because they're held back. And so you end up with this ladder. And so the ladder shows a band at every place that this enzyme can break the DNA, all the pieces that got broken at that location. And for other reasons, other methods I don't have time to talk about, they knew where along that length were these 3 sections called the operators OR 1, 2, and 3. They just labeled them there for us. And so what they want us to see is that when they put protein in that, the repressor protein in increasing concentrations at the right-hand side in lane 8 is the really high concentration. There's no bands in this area. The DNA is all protected because the protein was sticking there. And because they diluted it to find out how much protein you have to accomplish that, they can measure sort of how tight it's sticking, and they notice that you only need a little bit of protein to start protecting these guys, but you need a huge amount of protein before you start protecting this guy. And that fact became the beginning of their mechanistic model of how this whole system works. Again, I'm not going to be able to walk through that. I just wanted to get the idea of here's an experiment that draws a conclusion that maybe a reproducibility project would want to come back and say, well, did they get it right or were they wrong? And in this paper, there are no p-values. There are no replicates. That doesn't mean this was not done more than once, because just to figure out how to get the right concentrations and stuff, there were all kinds of preparatory experiments. But the paper is highlighted here on a list of the other papers in that laboratory on this topic in the 20-year period from 1965, when they first identified that repressor protein, to 1985, when they pretty much finalized their model for how it worked. And so I'm just making the point, obviously don't read the titles, but that paper was built on the results of dozens of previous experiments, all of which had drawn some minute conclusions that influenced the model, and they were all implicitly tested by this experiment. So if any of those other assumptions had not been true, the experiment would have not worked out. And then it became the platform for dozens more experiments that tried to test other things. You know, if you mutated a certain way, does it break the way we think it should, and so forth. And at the end of that whole process, we end up with a model. So when I say model, you know, biologists working on trying to build models, I mean something like this. It's a very concrete, literal picture of physical things like protein and DNA, not abstractions or concepts like, I don't know, psychological tests and things often measured, but like an actual thing which has physically measurable properties. And it looks like a cartoonist just made this up and they could have drawn anything that they wanted, but it's not the case. Every single place that you see, for example, any two things touching, like that repressor dimer touching right there, that's justified by where the band was on that gel. And anywhere you see two proteins touching each other, like where it says repressor dimers up there, that's because another protein binding assay showed that those parts of those proteins bound, and so forth. So there's hundreds of experiments that go into coming up with a model. And this is sort of the end goal of the project. And it took lots of labs, lots of years to get there. And one of the points I think is important is that each conclusion that is believed to be a true fact in the system is supported by more than one kind of evidence. So we think this protein binds to that DNA because of the experiment I showed you, but we also think that because they crystallized the protein and got its three-dimensional crystal structure from X-ray crystallography and found the exact shapes of the parts of the protein and showed that they fit perfectly into the groove made by the DNA and so forth. So there's redundancy in the way people approach problems. And these are some of the principles that I think biologists are trained to figure out if their conclusions are right. It's not like run that gel 20 more times. It's like, well, if that's working the way you think it is, then this should also be true. Try that other thing. And I'll talk about why that might be. And then the final step and the final goal really is the hope that when they figure this all out, it'll turn out not to be unique. That other cases in biology will be similar because of evolutionary conservation. They won't all be the same, but you'll be able to find some other gene like this one in yeast, which is a eukaryote instead of a prokaryote, so a very different story with regard to what's going on with the DNA. And yet, even though this system is different, they were able to find the principles that they looked at in that system could also explain much of what was happening in this other system. And so this is sort of speaking to like what the goal of the work is to come up with these abstract concepts or general principles. And I don't know whether the people of philosophy and history of science have paid any attention to this book or this story, but Mark Patashny is the lab who I've been talking about, who is also my PhD thesis advisor, which is why I know the work pretty well. Although none of that was my work, that was all before my time. And in this book, it's a little pamphlet. He does a beautiful job, really self-consciously describing how we know that we know things. And so I think it's a really nice case study. And then again, the long-term goal was to come up with a picture that was not at all particular, but very general, and says every case is going to be different in some detail, but lots of them have a feature that there's some protein whose job it is to determine where a process happens, and it determines that because there's a code on the DNA that tells it where to go. So again, apologies if that was too fast for people who didn't know the story, but it gives you an idea of what an experiment is. And now, very quickly, completely different kind of story from visual neuroscience. And this has to do with the first few steps by which light entering your eyes makes its way to your brain so that you see things. And the diagram left is just to show you where we are in the brain. Light going in the eyes gets turned into an electrical signal in the retina, sent to a relay station in the middle of the brain, and then back to the occipital cortex, the back of the brain. And this is a drawing from Ramon Cajal, more than 100 years old. When I put this up, I realized I hope people are studying Ramon Cajal like crazy, because it's another gorgeous example of like how things are known. He filled individual neurons and individual specimens and traced them, and then after looking at hundreds or thousands of such examples, conceptually integrated them into this picture of the circuit and how he thought the circuit was connected up and worked. And I think pretty much every drawing he's ever did was right. He didn't draw everything that exists, but everything he drew, I think he got right, which is just phenomenal. But anyway, so this is this pathway from the eye to the first, when it first arrives in the visual cortex. And I'm going to enter this story again in the middle, in the 1960s, when Hubel and Wiesel did the famous experiments. Again, forgive me if this will go so fast. But it was already known that in the retina and in the LGA, the first two steps, if you found any one neuron and you stuck an electrode in it and you try to figure out what made that cell get excited, you flash lights all over the room, and everywhere that where you flash the light, the cell gets excited, you draw a plus. And everywhere that you flash the light that the cell gets silenced, you draw a triangle. And you find that in the entire 360-degree environment, there's only one place in the room, right there, where this cell pairs what's happening. And it likes to see light in some center area, and it prefers it to be dark around that. So center surround receptive field. And they found other ones that prefer it to be dark in the center of their receptive field and light around. So this is the setup for the experiment, where people Wiesel then march to the very next station. We know now how the brain codes the scene, right? It makes a little pixel image of it in these dots. So they go to B1 to find the dots, and they don't find them. They find that these cells respond to light along some elongated bar instead. So they weren't expecting that. It was a completely serendipitous finding. They wrote a paper describing that result. Again, no p-values whatsoever. Many cells that didn't match this, some of which they mention and some of which they don't. But they tell us that they saw these and they had this idea how it could work. That maybe that cell in visual cortex that they're reporting from, it's getting input from a bunch of cells in the LGN. And each one of those cells is responding to a circular dot. But their circular dots that they respond to happen to be lined up in a row. So if this cell is going to get excited when all these get excited at the same time, and that would be when a bar happens. So that was their model. Again, the parallel here. This is not an abstract theoretical model with sort of floating things like, I don't know, utility and happiness, something like that. It's physical cells with physical connections that have specific properties that have been measured. And they've made up one new hypothetical fact that would be able to explain the data that they found. And a long time, this wasn't really tested, this model. So much it was just carried forward as the working model for how we could understand things. And lots of other experiments were done that mostly remained consistent with it, so it didn't get updated until 30 years later, when I'm now blocking the names of Clay Riedenhuis and Manuel Alonso, were able to do the killer experiment where they put an electrode in the LGM recorded from one neuron, and then they found in the visual cortex the one B1 cell that neuron was directly connected to with the synapse. Okay, cool story how they did that, but nevertheless, they finally figured out how to do that so they could record from two connected cells at the same time. And here, instead of pluses and triangles, they use red and blue to indicate red where bright light excited the cell, blue where darkness excited the cell. And on the left is the raw data, and the right is just smooth. And it just shows, here's the LGN cell. It has sort of a circular receptor field. Here's the cortical cell. It has kind of an elongated receptive field. And since they were recorded at the same time, we could take the circle showing where the LGN was excited and superimpose it on the V1 receptor field. And sure enough, everywhere that if the LGN cell was responding to bright light, its receptor field always lined up with a bright part of the stripes in V1. And they did that for a bunch of cells. They did not know how many cells they were going to do when they started this experiment. They just kept doing them until they felt like it seemed pretty convincing. And what they came out with here is, almost every cell that responded to bright light in a circular spot was wired up to a cell, somewhere along the length of the stripe that stripy cell, stripe sensitive cell was responsive to, confirming that model. So there's a lot more to this story. There's a whole business called complex cells. People still fighting about details of this, But the point is that it kind of follows the idea of how one experiment fits into the middle of this big fractal process. And the models are these very, very literal concrete things, and they're sort of works in progress that evolve and get modified as new data come in. So general themes. The goal of the work is to come up with abstract principles and concepts. The individual facts that are measured are necessary to that, but I tend to think of them more like, often you'll see diagrams about, rigor in science, and they'll have some facts, and they'll show it like a cornerstone at the bottom of a tower, and you're going to put some other block on top of it and build some tower. So everything hinges on that block being right. And I think of it a lot more like Velcro. Like every specific claim in the literature is like one loop of a Velcro strip, right? And it's the hundreds or thousands of little loops and hooks together that are make the whole model plausible. And it wouldn't be good science if most of those loops weren't right, but it's not tremendously important if one or two of them turns out not to be. The observations are sort of cumulatively summarized in an updated working model. So it's not like we have a final answer and then we say we know X. And those are very concrete models, physical, causal, mechanists. They have objects, interactions, and processes. And they're also highly constrained. I didn't have time to talk about this too much, but there's all kinds of other outside knowledge that comes to bear. Like you can't draw a physical model of objects with two objects occupying the same space at the same time. Right, so you've got a hard constraint of steric hindrance that makes most of the cartoons you might want to draw simply not possible. So it's different, I think, from a lot of social political science models in which there's really no specific constraint preventing you from making up postulating, pretty much any model. And so when you get into those cases like those papers at the beginning, we're saying people just dredging through their data and just make up one model after the next until they find one that the data say are significant. But that'd be hard to do in a biology experiment because almost every model you'd make up would simply be ruled out as impossible. So you're pretty limited in the ones that you can even test. Right, there's this tight iterative loop, which I've been harping on, so I don't think I have to go into that anymore. And then the last thing is that the conclusions that the field accepts as true usually rest on multiple kinds of evidence. And I think that's actually a really, as I reflected on my experience in those two fields of science, I felt like that was maybe one of the big takeaways. And that is, I think I have more coming up on that, but I'm not trying to say that biologists get everything right. Like they get stuff wrong all the time. So we expect that we're going to get stuff wrong. And when we screw up, though, it's not usually, it can be in some circumstances, but in very many circumstances, it has nothing to do with we didn't check enough mice. It's more often our method has an assumption in it that isn't right. So if the whole DNA footprinting method premise was just wrong, then those results would not be reliable. But they'd be totally reproducible. You get the same gel every single time, but it would still be wrong. And so the worry that biologists, I think, lose sleep over more is, since my methods are indirect, I don't know for sure that they work the way I think they do, that they're really measuring what I want to know. And so the way you get around that is you try to measure the same thing in lots of different ways. And you figure they're probably not all going to be, broken and misleading in the same way. Right, there's this idea that the conclusions, I think, within a field are usually considered provisional, not final. They're graded and not binary. So people don't come up with a hypothesis and say true, false. They say, well, I'm getting really pretty confident of this one, and that one's still pretty flaky. They tend to be particular, not general. So one of the funny things biologists will say, and you'll see it in the papers, I don't know if anybody ever did one of those word analyses of it, is people will say such and such, in our hands, this drug causes this behavior. And people use that phrase because they know you can do the same experiment in your lab, you might get a different result. But that's okay, because I'm just saying I have a system in which that pertains, and I can leverage that to make an inference about the model. And the models are approximate, not exact. So often there's a factor or two that don't fit, and people have room for, well, there's a weird thing going on there, we'll figure out later.
And there's another thing, which is that, in biology, readers are expected to judge every paper on their own and form their own opinion. And it's very much expected that people will disagree. You have a journal club. I mean, it's one of the main ways you train PhD students in biology is they have classes. And then after that, for the rest of their careers, they have journal clubs. New paper comes out, 8 PhDs sit around a table and decide whether or not they believe it. And they'll disagree. And one will say, yeah, I don't buy it because of this. And somebody else will say, no, I totally think they're right. And so it's naive to think that somebody who's not even in the field could come in from the outside and pick up a paper and expect that the field understands this to be a perceived truth. It's not. The field receives it as a contribution, like some evidence and some arguments to be considered. And even when you look at one figure in a paper, it's not an experiment. I'm told that in some fields of the kind that were first being critiqued by the reproducibility crisis narrative, often will be reporting the result of literally one experiment. And I'm not sure they all realize that that's not what is happening in biology papers. And established knowledge evolves over lots of papers and labs and years. Okay, so one thing that may be conspicuous is that there's very little talk of statistics in all of this work. So this is Fisher, and the Fisherians, I know will say this. So if this is science, where's the p-value? So it's not science unless you've done statistics. And the average biologist who I interview has pretty much this response. I have no need for that. I'll give them to you if you want them, but they don't convince me. I'll believe something even if the p-value is bad, or I'll refuse to believe it even if the p-value is great, because I have other reasons for what I believe. So what are some reasons that biology's gotten away with that? In some fields of biology, some phenomena really have very low variability and very large effects. So like that binding gel I showed you, it's kind of a black and white difference. And it's, if you just did the same gel 6 times, you wouldn't be able to tell them apart. So they can be wrong, but not because they're variable. That's one reason I think biology has gotten away without having, it is statistical reasoning to say, I believe this affects because it's big and the variability is low. That is statistical reasoning, but you don't have to do a test and put a number on it. I think I'm going to skip over some of these things because I want to get to some other points. So in these informal interviews I've been asking people, so what does go into deciding what you're going to believe? And there's going to be a lot of things on here. So I'm just going to kind of go through them by way of sort of throwing out there what kind of discussions these sound like. So one of the things that I hear a lot is the support for the model before the study began. Right? If there was like the Hubel and Wiesel model when Sayreed sat down to do that experiment, there was already a ton of evidence consistent with the model, and that influenced, the plausibility of the conclusion. And that's something that the null hypothesis significance testing doesn't really give you a good way to put in there. That would be a prior if you were doing Bayesian statistics. People talked about the amount and quality and consistency of the data. That's more along the lines of what the p-value helps you measure. The completeness of all the internal experimental controls. Did you try every possible combination of leaving each possible thing out of the tube in every combination and make sure that all the different variants worked out the way you thought they should? And then they bring in other information that they have their background knowledge, their sort of domain-specific background knowledge. They have read other literature, they've had other observations of their own, and they say, does this conclusion fit with everything else I know? And that's part of why everybody comes up with a different judgment of whether they believe it, is they're bringing in different background knowledge. People consider whether the experimental method they use is more or less direct or indirect. They believe an X-ray crystal more than they'll believe a DNA footprint because it's more, I don't know, a better method. There's theoretical constraints. If there are constraints like steric, if 2 things can't be in the same place at the same time, they want those to be met. If those constraints are severe, then the fact that a model can meet them all is considered strong evidence by itself. And then this idea of convergence, if the same thing was shown by 2 or 3 different ways. A criterion that people do invoke a lot is expectations that things should vary smoothly or monotonically. If you change the concentration of something, the effect should get bigger and bigger. Maybe it's U-shaped, it'll get bigger, bigger, and smaller, but if it's going up and down all over the place, they don't believe it. right? So believability, they have a prior that phenomena vary continuously, and so when things are not smooth or monotonic or relatively simple functions, they get more suspicious. They want to know if this is a biology specific thing, this mechanism you're telling me about, is it even plausible that that could be advantageous or that it would be selected for? Not much work is done by most biologists to show whether that is the selective pressure that led to that trait. Most people don't know that, but at least they would like it to, they feel more convinced that some model is right if they say, oh, I can see why that would be advantageous to the animal. And also kind of related to evolution, the idea, well, I've seen mechanisms that look like that before, so there's some precedent or some analogy or homology to some other system. And if a model can explain a lot of observations, it's considered more likely to be true, even if there are some exceptions it can't explain. And whether or not this is a good criterion, we can debate, but a lot of people do feel more compelled by a model if they feel it's parsimonious or simple. If it's got lots of moving parts, they're more suspicious than if it's a simple model. Okay, so I just wanted to end on going back to the whole reproducibility thing. So these headlines hit in December. This cancer biology reproducibility project, in which they picked a whole bunch of results from cancer research journals, and they tried to reproduce them, and they failed to reproduce a lot of them, and they actually failed to even attempt to reproduce most of them. And the headlines make it sound like most cancer research is crap. But the people who are doing cancer research and the people who are being cured by new cancer cures don't see it that way. So what's going on here? And I think part of the problem is summarized by this cartoon here. It's a bunch of surgeons around a patient saying, yeah, that's pretty much what it looks like on YouTube. Meaning the idea that people expect that without any specialized training or expertise, that they could just waltz in and do something like replicate some very difficult technical experiment. And so I think these reproducibility projects have seem to have had a lot of success and value in political science and social psychology. If you don't think so, I could hear that argument. But in any case, in biology, I think one of the reasons they're going to be challenged is that you need expertise just to interpret the literature and figure out what it is the community believes to be true. The experiments are way more expensive and time-consuming than these people realized. They had like a $1 million budget, and they were going to do like, I don't know, $100 million worth of experiments. They just had no idea. The experimental training required to be able to get the techniques to work takes a long time. The people who do these experiments train for 5 or 10 years at the bench next to experts to learn these techniques. It costs a lot to train these people. And the people who be in the field do train these people. That's why they take on PhD students and postdocs. But they have to be super selective, right? They can only do a few, and they take people on that they think have the aptitude and the determination and so forth. And resources are very scarce. People work really long hours, and they try and get by on really tight budgets. So if someone calls you up out of the blue and says, we want to just try and reproduce that experiment that you published last year, as a test to see if it's reproducible, and we want you to help us, and we want to come to your lab and you're going to train us how to do it, you can understand it's not just people being stubborn and obfuscating the process to say, you know what, we don't have the resources to do that. If we thought that was the most informative thing to do with our lab time, we would be doing it, but we think it's more informative to do some other experiment that will test the model from some other direction, say, than to repeat that one. So it's true that a lot of people did not cooperate, but I wonder how much of it was just naivete as to like how hard that cooperation would have been. And then there's this problem, which I do think that biologists have some guilt for. They put the p-values in the papers. They say this isn't a significant effect, p whatever. So the reader isn't justified in thinking that if they see a value p less than 0.05, that should reflect something that they can try and verify. It's not at all trivial to get from there to what should be the probability that that result would reproduce, because that's certainly not the p-value. But we did give them a false lead by telling them a p-value, which wasn't our real reason. And finally, it's impossible, there's a lot of calls for it. The problem here is they didn't put all the conditions in the paper necessary to repeat the experiment. And it's impossible to do that. And I wrote this down, and I thought people are going to say, oh, it's not impossible, you're just being lazy. Just write a really, really long supplementary materials and tell us everything. But I'll stand by it, it's literally impossible. We have no way of knowing what conditions could possibly be influencing our output. So what we do is we summarize the ones we know matter, the ones we think might matter. And beyond that, if it turned out that the time of year that we did the study mattered, or the gender of the animal trainer, or something else that we didn't think of, if it turns out that that matters, it will be found out because somebody somewhere else will try to do the experiment and get a different result, and we'll pool those observations in that big fractal circuit, and we'll track it down. So that's why these sort of outside replication efforts, I think, are not likely to go well in biology. And so here's just a couple of ideas for where to go from here. So if we have this research field, and the literature is really an efficient way for these people to communicate among each other. So they won't mention, for example, parts of the methods that are absolutely standard that everyone in that field should know. They won't bother to tell you that their surgery was done sterilely. Of course it was. And so an outsider who doesn't know what's obvious and what's not isn't going to be able to know that. And that's, I think people who are trying to look at our fields from the outside and sum up how they've been doing, might want to look more at things like review articles or textbooks or methods manuals, which is where the field tries to codify in one place. Okay, we're going to sum up what the whole field thinks, or try to write out exactly how to do this experiment. And these are sort of intended to be self-sufficient in a way that papers are not. And then just as far as, the mea culpas here, as far as biologists, I think we totally support this myth. Null hypothesis significance testing is the only formal method that biology students are ever taught. If at most they have a statistics class where they're taught how to compute some p-values. And it's the only one that's ever advertised when we talk to the outside world about how we know things. Journals and funding agencies increasingly demand that your paper be written in a hypothesis testing narrative, even if that hypothesis is one you came up with after you saw all the data. They require that you give them null hypothesis significance test statistics, even if there was no prospective study design, which renders those invalid. And so what researchers do is they provide these perfunctory calculations that say, you want a power analysis? I'll give you a power analysis. Yeah, sure. You want a p-value? Here it is. And I will say, because their education was so poor, I don't think most biologists knew that those were lies. They weren't trying to deceive the world into thinking that they did a proper study. They just don't think that these numbers mean anything. And so they've given to you without realizing it. And they got to stop doing that. They have to understand they mean something very specific, and that it's misleading to give them wrong. And then there's this other thing, that these researcher degrees of freedom, this fluid changing of your experiments and your hypotheses and your analysis it's rampant. It's not the exception, it's the rule, which makes NHSTs not valid, but it doesn't make the research not valid. So I'm sympathetic to, actually, this is one of the authors of the first paper I showed. I have this is on this paper. The idea that one of the biggest things we could do for reproducibility and credibility in biology research is simply realize that most things are not null hypothesis tests and be upfront about that. And since I know I've gone over time, I'm just going to kind of go through my take-home messages. We got to stop faking that we're doing NHST. But unlike most of the reform movement, I don't think the solution is to start doing NHST properly. I think that would kill the process. I think what we need is to have more support in the funding agencies and the journals for openly non-confirmatory research, which I think is what almost all academic biologists are doing. There's room to engage with alternative statistical methods, exploratory data analysis, Bayesian statistics, visualization, and so forth, that are perfectly compatible with that workflow. These questionable research practices are not a problem with the experiment, they're a problem with the p-value. So if we didn't use p-values, we could use other statistics that would allow people to measure our certainty. But this is the one I think is most interesting from a philosophy of science point of view is maybe you guys have done it already, but we haven't heard the news of codifying what are these heuristic methods that we are using to draw inferences, to choose experiments, decide what experiment is the most informative one to do next, or decide whether a model should be believed. Because I think if we could talk about them in a more formal, organized way, it'd be better, easier to teach them and easier to communicate like the real reasons behind our conclusions. And then finally, identify and focus on the ways that we really fail. So I don't think it's usually because of sampling. It's because of something else like assumptions. And there's a couple of things that these are just examples of failure modes of really nice papers that use simulations to show why analyses that biologists find convincing can fool you entirely. Biologists would believe that this is correlated because it looks smooth and continuous and there's noise that, it's much more than the trend, but it's garbage. Or people will believe, that this result is true, that neurons in the mouse brain encode cryptocurrency prices. And they believe it because there's the control and it's a big effect, right? But it's because this analysis is invalid. So that kind of work, using simulations to show the flaws in people's logic is a helpful way forward. And then there's these projects where people actually get together from within the field and say, we need to know how reproducible these things are. Let's all get together. Let's 10 labs agree that we want to know whether you could train a mouse that a decision-making task and get the same results in 7 different labs. And this is just one figure from a paper in which this big collaboration had all agreed to do the exact same experiment so that they could measure the reproducibility of things. And here's another one like that for a drug assay study. And these projects are hugely informative. They're incredibly costly. And they don't sort of advance new knowledge directly. And so there sort of needs to be another funding stream and maybe even another professionalized workforce, who do these kind of things. So I'll end there. Thank you for your patience. I threw a ton of information at you, but I'm happy to chat about anything afterwards.
Okay, so we have the capacity for questions here and questions online. So for folks who are online, just so that I don't miss you, if you can use the raise hand function. If you don't want to ask the question in person, you want to just submit it in the chat, I can read that out. But the floor is open for questions for Pamela. We'll start with David.
Wonderful talk. I kept coming to preach, sister, preach. But I have a comment because I want to push back on something you said.
Okay.
And I don't think you'll disagree with me on this, but I want to comment on it. And then I have a question.
Okay.
So my comment is you keep talking about concrete physical things. And I think what you really mean, I will put words in your mouth. I'm going to say that the real key is constraints that are identifiable. So there are, I think, a lot of really good examples of non-concrete things that have been models on large scales. Reinforcement learning is the classic example. That is not a physical thing in any way, shape, or form, but has all of the properties that you're talking about that has allowed people to go in and really identify that exact model, that beautiful observed model experiment cycle.
Yeah, I like that generalization a lot. I think the history of biology coming from these very physical causal models is quite influenced how we work, but then the generalization to abstract models that also share, like you said, strong constraints.
I think the behavioral neuroscience world, which I know you've done work in as well, doesn't have that problem. That also lives in that same space.
Yes, I agree with that generalization.
So my question, though, is because I agree 100% with your point about the p-value problem, which I have hit. I mean, I say in my lab, I say statistics are BS. We don't believe any of this stuff. But you can't publish without it. So what is the truth separation paradigm that we are going to use? Because what you described is essentially consensus agreement And I
Well-
don't think that's good enough at some level.
Yeah, exactly, and that's where I think, I have hopes for, these 2 points here. You know, so one of the reasons I'm talking with statisticians and especially people in Bayesian statistics is, you know, there are measures of, you know, the strength of evidence for and against a hypothesis that do not depend in any way on how you set up your experiment, that allow for the evidence to be coming from qualitatively different kinds of experiments and so forth. So there are formal systems that are actually older than Fisher's methods for measuring evidential strength that are compatible with our workflow.
But like the Bayesian statistics have been almost impossible to actually make work in practice.
Well, let's talk offline about what you mean by that, but I don't know what small toolkit of statistics, you know, could replace the p-values or augment them in a way that would really move the field forward, but I'm hoping that there may be such a set. You know, I don't know, log likelihood ratio between 2 compared hypotheses or something like that, that have enough generality and strong properties that people could use them. So that we have other formal and quantifiable ways to measure evidence. I'll just push back on the consensus thing because in that big fractal diagram there's always lots of models.
Yeah, no, I
Yeah,
don't mean
so-
in the sense of final consensus, but
Yeah.
in the sense of you've got a bunch of models and everybody's arguing about them and eventually they kind of, you know, percolate to some level.
Yeah, so my experience and my introspection on that is what happens is certain aspects of those, in the beginning, the models would be really different. In the end, there's still people staunchly disagreeing with each other with different models, but there's increasingly a handful of characteristics that those models all have in common. And so then we can say, oh, those characteristics that everybody's model has in common, that's the part that we've settled. And the ways in which they're different, that's the stuff we're still hashing out.
I agree completely.
But yeah, and then this other thing is, again, I feel like there is a method to the madness of these heuristic reasonings. One thing I forgot to say is this idea of making decisions, right? So p-values, especially the significance test, was supposed to be a way to make a binary decision, you know, true, false, as opposed to just a graded measure of something. And the only binary decisions that I see people making in my research world are decisions about what experiment to do next. Those are the decisions that they're making on the basis of data. The models, they're not really decisions. They're sort of evolving worldviews, if you will. But anyway, so I think that if we had some way of talking formally about how we're bringing in different kinds of evidence, background information, or you know, theoretical constraints, then it might seem less loosey-goosey. The stuff that we now just narrate in our discussions sections, it's too verbose, basically, and chaotic for people to put a finger on, like, how good is that reasoning. But you know, there's a history of people figuring out formal rules for inference that maybe we should be tapping into. And one of my agendas for doing all this research this year is to think about whether we should be teaching graduate students, you know, some completely different arsenal of tools for thinking about these problems. You know, maybe they don't need to learn 8 parametric and 8 non-parametric hypothesis tests. Maybe they do better to learn symbolic logic, or I don't know, something else that might be more useful. So that's what I'm kind of trying to find out.
All right, we've got one on chat from Ron Okagaki. He asks, will standardizing protocols, procedures create more problems? Several labs using the exact same protocol risks systematic error, but if protocols vary, might results be more robust?
Yes, I think I'm on the same page. I don't think it's a good idea for the entire field to become standardized. I think occasional efforts, if there's something that a field does a whole lot of, to have a test run and find out about, you know, the variability is a good thing to explore. But I think for exactly the reason I was talking about converging evidence. You know, so I'm doing animal behavior now. We do decision making. Lots of labs do decision making. I visited 8 labs. You know, some of them, they have a mouse whose head is fixed. and they're pressing levers, and others is mice who are running around in a box, and sometimes it's rats, and sometimes it's monkeys, and there's a lot of diversity there. I think that's necessary because when we are converging on similar views, that's part of what addresses the problem, that we assume anchoring a mouse in place doesn't affect its decision making, but that's an assumption. Maybe it does. So we don't want everybody doing that. So I think I'm agreeing.
So let me follow that up with a question that's a sort of variation on the theme. So this emphasized protocols and procedures, but in your sort of fractal space, you have more than the experiments. You have, the models, and it seems like maybe one of the things that's also important in thinking about what kind of variation is maintained in the population of scientists also is with respect to models and other sorts of things, that basically you don't just want variation in any one dimension, you actually want something like a system that is good at maintaining a balance between certain degrees of consensus and certain variation in not everybody being convinced.
Exactly. I realize this is going to be our new buzzword. We need biodiversity to have a stable research ecosystem.
Okay, Ruth. You're muted, Ruth. Let's get you in.
Trying. Okay. Yeah, thank you for such an interesting talk. I think you said toward the end, sort of acknowledged that one aspect of the work you were describing was that most of the results that sort of feed into the models are very clear cut. They're sort of binary, discrete, major effect kinds of things. And I think that's characteristic of molecular cell biology. I am not in that realm. I wonder to what extent you've thought about, well, how much your view that you present here is restricted to those realms of biology, or any comment you'd like to make on that.
Yeah, absolutely. So I think that part of why the history of the field of biology has neglected statistics is because it began with experiments like phage experiments where N was infinity and results were black and white. And so those quantitative variation wasn't the main concern, but that's obviously not true for, in fact, Mendel and Darwin, maybe they put them up there with Monica Hall as the greatest biologist of history. They're concerned entirely with variability. There wouldn't be anything to talk about if they weren't dealing with variability and measuring that. So I don't mean to say that we don't have small effect sizes in variable populations, but I think there's a history of the field that grew up through the molecular biology era thinking that way. And that could actually be a problem. There's a lot of people now who do behavioral neuroscience who got trained in molecular biology, like myself, and we may be treating animal behavior experiments as if they were molecular biology experiments when they're really not. And so that, that's a thing to be cautious about. So I didn't mean to say that we never have to deal with that problem. We often do have to deal with that problem. But the cases that were not like that, I think, have influenced the development of the field's methods. Does that answer your question?
Yes, thank you.
All right, got a couple questions here. So let's go in Lauren, Steve, and then Yoshi. Lauren, go ahead.
Yeah, so I think my question actually builds nicely off of Bruce. In that I noticed that the two models that you presented were both mechanistic models, where you're using various data points to put together this cohesive picture, which I guess I have some reservations about the idea of concrete or facts or kind of a plain assessment model. I think there's decision making that goes into compiling general constructive models, but I guess I'm wondering about how much we can generalize removing p-values from all sorts of different biological sciences. In that, like, I think about times in molecular labs, and then I think about times working with Software where I'm interested in like differences in different species behavior on like preferences. It seems like there I'm leaning more heavily on p-values and statistics to back up my observation in a way that I don't when I'm working on kind of a molecular mechanism.
Yes, so I tried to cover that with my caveat in the beginning that I'm not covering all kinds of biology because there's so many kinds. I think that, not that we should abandon statistics. I don't think that p-values measure what we want to know. I think people want to know how likely is it that the result that I got is real. And that's the PPV, not the p-value. And I realize it's hard to measure that, but I think that's what we want to know. So I think p-value, people think that they're measuring that with p-value and they're not. So I don't think p-value is the right statistic, but I think we are going to need statistics to deal with those kind of cases. I guess I just want there to be room in the field, you know, at the funding level, the publishing level and so forth, for that. Not, there are fields where that's not the kind of reasoning and it's more pluralistic. So I'm just sort of focusing on the part of what I think that's controversial, but I don't want to banish statistics, maybe p-values.
Steve.
I think mine's a follow-up to what Lauren just said. So that you have the p-value. I mean, I'm a family physician, so I'm not a biologist or any of this stuff, but so it's the probability that you would get the answer you got or more extreme if the two groups were the same. When you were just, at least that's the definition I use, feel free to tweak it, but the examples you used, Lauren used the term mechanistic, I would use the term descriptive. And I don't know anybody, I'm unaware of people who are arguing that you should use p-values in descriptive research.
Thank you. The journal article, well, the journal editors won't let us get to it.
That one of the consequences, I think, of the alarm that's been set by the reproducibility crisis narrative is that everything everybody's publishing is wrong, and the journalists think the way we're going to solve this is ask everybody for their p-values, and then now is another layer saying, oh, well, those p-values won't be valid unless they were pre-registered. So now we want to ask everybody to pre-register and give us p-values, and that will provide a very rigorous measurement of that thing that you just said. And by the way, kudos. There are very few medical doctors who can define a p-value.
I mean, there's journals, there's lots of calls to get rid of the p-value. There's some journals that won't accept the p-value. You can argue with the editors to get things in, maybe you are in different journals, but I'm just not aware of these terms.
Yeah, I think there's, I was perceiving a direction towards increasingly strict adherence to p-values. And, you know, what part of why I got involved in this is I just said, well, that doesn't apply to my research. If we want my research to be rigorous, we should have some other things on that checklist that could be a p-value or this or that, because it's addressing the needs of a particular kind of experiment or a particular kind of science, but it's not all of biology.
So I would just jump in really quick and just following up on what David said, that it's changes in some of the publishing expectations that people pick up on, and there's now these checklists and things that have to be completed just to submit the article. And it's those things that have made people more salient about why are you asking me these questions. But the push to ask those questions is related to this broader concern that if we don't ask those questions, somehow the research is not going to be sufficiently rigorous in some dimension. But it doesn't necessarily ask, should these questions be asked rather than these questions? And that I think is part of what you're doing.
And some journals have better checklists than others. I mean, there's apparently it's now official, there's the statistic wars, and depending on who, you know, which side of that battle is in sway with the journal, you know, they can be very anti-p-value or absolutely only p-values or anything in between. But what scares me, and I think is a problem, is all the biologists who don't really think it's important, and so they're willing to just throw some fake p-values in to appease people. That's terrible. That's terrible. You can't do that. But that's what I think people are doing, and they need to understand that that's not the solution. this might be a slightly off analogy, but I feel like it's sort of like Galileo trying to justify, his heliocentric theory to the Pope by some Bible quotes. It's like, you want some Bible quotes? I'll give you Bible quotes, but that's not how he drew the conclusion. And he's not prepared to go to the mat with whatever the Bible says. So he shouldn't be pretending that that's his evidence, So I just think that in my community, I think, people need other answers that they can give to why I know this is rigorous. If they're not prepared to give a p-value that's legit, and I don't think they're able or willing to do their science the way they would have to for it to be legit.
Okay, so Yoshi.
Yeah, so my question is related to some of the previous questions. So you mentioned low variability in this basic experimental biology.
In some kinds of experiments, yeah.
Yeah, and I believe I understand correctly. So I think you mean by that something like researchers know that like in these fields, like those fields of basic experimental biology, they can expect low variability. Maybe
yeah, I think it's very domain specific. So I interviewed a few people from like classical molecular biology and Matt Messelson and people like that. And every single one of them said a sentence almost exactly verbatim, like, if you need a statistical test, you've designed a bad experiment. If you've designed a good experiment, the answer is black and white, yes and no. You don't put the enzyme in the tube, nothing happens. You do put the enzyme in the tube, it happens. And if it's some blurry quantitative in-between thing, you should think of a better experiment. And that isn't going to work for other fields, but that's a whole field where that.
Yeah, good. So I think I understood correctly. So my question is, yeah, like responding to some earlier questions, you said like some fields of science might need not maybe like strictly p-value methods, but like something like that, some statistical methods like that. So I think we want to know what factors make the difference. And I think about basic experimental biology, people are talking about things like the availability of standardized experimental systems, or like things like model organisms, availability of experimental controls, and maybe, I don't know, like the type of biological phenomena tended to have that kind of like less variability. But I want to hear from you, like, what factors are relevant.
Yeah, I wish I had the answer now, but I do think that's one of the things that's got to be on the to-do list for this agenda is, if we're going to say we've got a pluralistic set of ways of inference, and some of them are statistical and some of them are other, we also need a way to be able to classify a particular experiment as belonging to one of those groups. And offhand, it seems to me the main thing that makes something fall into the category where you really need a p-value or some other thing that's directly measuring, variability and noise and all of that, are cases where the effect sizes are not huge compared to the variability on repeat, right? And so that's why I said, I told my statistics friends with these all or nothing experiments, I said, they don't need statistics. And they said, yes, they do. They're doing statistics, but it's just that, if your effect size is 10 to the 10th, and your variability is, 10 to the 2, it isn't necessary to do the math. They've done statistics. They've just done it, on the back of an envelope. So, and then there's things that are clearly so overlapping, where you're seeing a 5 or 10% difference in an animal's behavior because of some manipulation, where you desperately need to use statistics, or you'll fool yourself every time. And then the only reason I'm harping on p-values is that I think for some experiments, you could just do the whole thing over again tomorrow. If it's a phage cross, you just do it again tomorrow. But if you're doing some longitudinal behavioral study in rats or something, it takes you 2 or 3 years. you can't keep starting over every time something changes. You have to be able to deal with the fact that you changed your hypotheses, you changed your methods, you changed your analysis all along the way, and find a way to harvest all you learn from all those data in your statistics. So the p-value is just not a good statistic for that, for that situation. But yeah, I don't know if there's going to be other criteria for classifying what kinds of inferential methods are valid for what kinds of scientific problems. Yeah.
I think one of the criteria that's really important is data rich versus data limited. And a lot of the fields you're talking about, and your example of the debate was a great example of that, is a data rich world where there are dozens of different ways of looking at the data. I mean, when I go in my lab, we record from huge populations of neurons, and we can look at it 26 different ways. So if the hypothesis says this is true, we can come in, you mentioned it's coming in a different direction. A lot of the places that the real reproducibility problems have been hitting in, say, social psychology and things like that, they actually have very limited data collection availability currently. And so they're coming in with what is effectively a single survey and things like that. And that seems to me to be one of the big crisis kind of examples where things are not showing up as reproducible. Because it's, if you have the multiple dimensions to look at, you don't have to use that simple statistical question.
Right. Yeah. I think that's right. Although it raises another point that I glossed over completely because I had way too much already, which was the multiple comparison problem. Because, when you get rich data, you have this other problem that you can fish.
Well, but I want to be careful. I'm not suggesting that on rich, yes, there is a rich data on fishing, although the truth is, in my experience, the fishing has always come up to be correct, just as a point on the rich data. But the reason is that fishing comes up to be correct because when you find it in one dimension, you test it in a different dimension on your data.
If you can, yeah.
Well, that's my point. That's what I'm saying. Is with rich data, you can do that. Whereas with the limited data streams, you have multiple comparisons, you run into trouble because you don't have a way of validating. And I think that validation is something that is underappreciated in most of the fields right now. When you look at things like the behavioral neuroscience, where things are in fact quite reproducible.
Yeah, so I guess that comes back to the question of what is the purpose of an experiment? And a lot of experiments that we do are really hypothesis generating. They aren't the end of a study. They don't give us a conclusion, but they tell us where to look, where to then go in with some other approach.
I like your model example. I mean, I don't think it's really, I think that, in fact, I would push back against the idea of hypothesis generating versus hypothesis testing.
Yeah.
I would actually say that's the wrong, that's a Fisherian statistical dichotomy, right? Whereas what you actually started with was this beautiful practical description when I'm looking like, yeah, that's how I work, right? You have a model which you're kind of building. You build some experiment to test some piece of the model. The experiment shows you a novel thing. That changes your observation. You build that changes your model, and then you go back to that same experiment, looking at it in a different light, and you can test a different observation. You essentially do a new experiment on the same data.
Yeah, data reanalysis is another thing, which the Fisherians don't have a great way to.
Well, they don't, because they're building on the hypothesis generation, hypothesis testing.
Right.
But your model cycle totally believes it.
Right. But yeah, okay. I know we've run late.
Yeah, so we are out of time, but obviously this conversation has been great. I thank everybody for joining us. We had people online, people in the room. This was a little bit of an experiment to see how the hybrid format would go. I think it was successful. So thank you, Pamela, for the talk. We really appreciate it. And again, reminding those who are there online as well that there are activities tomorrow. The biology interest group in the morning and then lunch. Please let us know if you have any questions about that. Otherwise, everybody take care and have a good evening.